Oh. Welcome back, class. This is week 12, so the last week of semester, and great stuff to all the students that have managed to get online tonight, because I know how busy everyone is. This is can be a very stressful time, the end of semester, stressful for students trying to complete assessments, stressful for staff trying to get all the assessments marked and calibrated and approved before returning them to students with their marks. So well done on attending tonight, for being able to attend tonight. So we've got 2 lectures to get through tonight, our final lectures, and we're going to start here with a lecture on taxonomy. Right, so let's start with a look at classification. Classification involves the grouping of organisms with common characters, and this actually performs a very essential function, or several functions in fact. This is important for the purpose of communication, for the purpose of information storage, retrieval of information. So if you're communicating with someone about a particular group of organisms, you don't have to list all the characters that are known to be associated with that group of organisms. You can just use words such as marsupial or mammal or angiosperm, and each of those words actually is associated with a very specific set of common characters that link those organisms within that group. So it's a very efficient way of transmitting information, but classification actually does more than that. It actually serves to predict new information. So imagine if there's a group of organisms that share, say, 5 different characters, and you find another organism that has a couple of those characters, then you can actually predict that they might also have, for example, character number 3, 4 or 5. And here's an example actually shown here in these photos. So studies on the Moreton Bay chestnut found that the seeds are poisonous because they can contain an alkaloid, a specific chemical. And they found in studies that this chemical controls cancer cells in rats and also inhibits one of the proteins of the AIDS virus. So they went on a search for other sources, and they used this classification system, and they found that this particular plant in South America has very, very similar, a very distinct pollen to this Moreton Bay chestnut. And sure enough, it actually contained that same chemical that's been studied in the research labs. So classification is studied by biologists called evolutionary biologists, and they basically aim to group organisms according to a pattern of common ancestry. And this is termed phylogeny. And these biologists, they actually work in the field called systematics. So they actually discover trees of life. Just like your family tree, that's what they basically construct, but they call them trees of life. And classification is very dynamic, it's changing constantly. All right, so here's one of those trees of life, which is called in the area of classification, it's referred to as a phylogenetic tree or a cladogram. Yeah, I know different words for the same thing, but you can see that this appears to be a tree that's lying down on its side. It's said to be an unrooted tree. Unrooted. There's no base of the tree, or rather the base isn't shown, so we can't actually see the primitive ancestors of any of these groups of organisms, but we can see how closely related they are to each other. So each group of organisms is allocated a separate branch, as you can see, and the closer the branches are together, then the more related those groups of organisms are to each other. And they actually constructed this tree by taking the sequences of ribosomal RNA genes that we talked about last week. They took the sequences of those genes, that is the order of the nucleotides within those genes, and then they aligned them with the sequences of ribosomal RNA genes in other organisms. And the more closely aligned they were, meant that the more closely related those groups of organisms were. And that's what determined where these branches are on this particular tree. So we can look at some examples. So here to the right, we've got this dark green branch, which represents the land plants. And you can see that the closest branch is the green algae, which belongs to this kingdom, the protists. And of course, they are known to be the most closely related to each other. Then we've got down here, we've got these branches, which represent our prokaryotes, bacteria and archaea. We've got the animals in blue, and in yellow we have all the protists. And this kingdom, the protists, this is really just a collection of groups of organisms that don't really fit anywhere else. So traditionally they've just been thrown into this particular kingdom. And we can see that the closest relative to the animals is this particular branch of the protists called the choanoflagellates. And yeah, which is quite interesting, given that choanoflagellates, these are mostly single-celled marine organisms, but they tend to have ribosomal RNA genes that are very, very similar to those found in animals. Okay, and here's another example of phylogenetic tree, and this one is does have a base. It is rooted, and we can see the common ancestor of all these different fish here in the bony fishes. Okay, so the study of methods and principles of classification that basically govern the naming of all organisms. This is called taxonomy. And basically, since the time of a very famous Swedish naturalist called Carl Linnaeus in the 1700s, the system of classification has used a hierarchy of different groups. There are set rules, international rules for naming organisms, and we'll look at some of these rules a little bit later on in this lecture. Okay, so the hierarchy of groups that are used are referred to as taxonomic levels, where each taxon is basically a level or rank of classification. Everyone's familiar with the term kingdom, so this is basically sits at the top of the pyramid. It's the highest level, and it's the most inclusive taxonomic level because it includes everything below it. Whereas if you go to the bottom of the pyramid, you'll have the species level, lowest taxonomic level, and it's the least inclusive because the species level only refers to a very specific species. So let's look at the taxonomic levels on the next slide. Here we go. So here's the taxonomic hierarchy. Highest level is kingdom. So for example, kingdom Animalia, kingdom Plantae, and so on. So beneath the level of kingdom, we have several phyla. So just note that phylum is singular, phyla is plural. Then under phylum, we have class, then order, family, genus, and then finally species. There are some intermediate levels here, and they're indicated by the prefix sub, so subphylum, subfamily, subspecies. All right, just note that species, the word species can be used for both in the singular and in the plural. Rather than just saying specie, one species, we still say one species. That's the generally accepted pronunciation for that. Okay. All right, so this is a good one for you to remember for your final exam, to actually be familiar with these different levels and where they sit. And now let's look at some examples. So for viticulture students, we have the grapevine, which of course is found in the kingdom Plantae. There are, I believe, 12, from memory, 12 different phyla under kingdom. Oh. Okay. So there's about 12 phyla that sit under kingdom Plantae. This particular phylum is Magnoliophyta, which includes all the flowering plants. Class Magnoliopsida, which are all the dicotyledonous plants or dicots. Order Vitale, family Vitaceae, genus Vitis, and species. So this is the common grapevine, has the species name Vitis vinifera. Now, just note from this also that at each taxonomic level, we have a single word name until we hit the species level. Then there are two words that represent the species name. Also note that when you're writing, when you're using a species name, make sure it's always in italics, and make sure the first part of the species name is capitalised. The second part is always lowercase. So make sure you observe that in all your future assessments. Also note that the first word from the species name is taken from the name of the genus. So that's our grapevine. Okay, here's one for the equine students, the horse. So we've got kingdom Animalia, phylum Chordata. There's about 35 phyla that sit under kingdom Animalia. Class Mammalia, order Perissodactyla, family Equidae, genus Equus, and species Equus ferus. And in this case, we have subspecies, which refers to the domestic horse. And of course, this distinguishes the domestic horse from various species of zebra, donkeys, and Przewalski's horse, and so on. Okay, so let's look at just some of the rules that are used in classification. So these include the endings of animal family names. So the ending is always ide, whereas plant family names always end in aci. And you can see that on the previous two slides. All the taxonomic levels, except for the species level, as I said, have a single word name. And here are some examples. Only the species name is identified by two words. The first word, of course, is taken from the genus that it belongs to, and the second word is referred to as the specific epithet. And this is basically referred to as the binomial system. Binomial meaning two names. And this is a classification system that was basically developed by Carl Linnaeus, the father of taxonomy. So before Linnaeus actually developed this system, there were species names that were incredibly long. There could be 10 or more words within the name of a species. So it just wasn't practical. It wasn't a workable naming system. So this really simplifies everything in classification. So species names are composed of two parts. Remember the genus name. So the first word in the species name is always capitalised. The specific epithet or the second word in the species name is never capitalised. And that combination of the genus and specific epithet is unique for every species. Here are some more examples. Tachyglossus aculeatus, which is the echidna, Aquila audax, which is the wedge-tailed eagle, and Latoria nursuta, the rocket frog. All right, of course, there may be more than one species in a given genus. Here are some examples for you. Of course, all eagles, and we've already looked at this one, the wedge-tailed eagle. Aquila, by the way, is Latin for eagle. Some of the specific epithets can be quite descriptive. So chrysatos is actually ancient Greek for the word golden. And then here we have Aquila heliarcha for the imperial eagle. You might also see that that second part of the species name, the specific epithet, can be used in several very different, very unrelated genera. So we can see three very different animals here, not related at all. All, of course, belong to different genera, but the same specific epithet is being used. Nasuta, which actually means nose. All right, so how do you define a species? It actually sounds quite simple. It is very, very complex, and there's no one definition of a species that is used by all groups that work in the field of classification. There are quite a few different concepts. Here are just two concepts shown here. The biological species concept, which defines a species as being able to reproduce and produce viable offspring. So it's really based on reproductive isolation between different species. And of course, that applies to naturally occurring populations. And that's fine when you're classifying animals, and it is the preferred species concept used by zoologists and other animal scientists. It doesn't quite work very well for plants. So plant scientists tend to use this morphological species concept, where they actually look at the form, the actual form of the organism, that is the shape, the structure, and so on. That is, they look at the phenotype. They look at the phenotype, the observable characteristics, and they use those phenotypic differences to determine or delineate different species. All right, so let's take a look at kingdoms, the highest, most inclusive rank of classification. In the 1700s, Carl Linnaeus classified all organisms into only two kingdoms, vegetables and animals. Of course, we call them plants now, but at the time it was kingdom vegetables and kingdom animals. And they had actually a third kingdom at the time, but that was for minerals, not for living organisms. Then in the 1800s, a third kingdom was introduced. In the 1900s, a fourth kingdom, and today there are 6 major lineages recognised: the bacteria, the archaea, protista, plantae, fungi, and animalia. So let's take a closer look at the kingdoms and the different schemes of classification. So at the top here, we've got one system here that recognises 3 super kingdoms. So all the eukaryotes are placed into kingdom Eukarya, and then our prokaryotes are placed into these 2 super kingdoms, Archaea and Bacteria. This one here is quite popular. So the 5 kingdom system, where all the prokaryotes are placed into one kingdom called Monera, and then the eukaryotes have separate kingdoms. But let's look at the 6 kingdom system. So prokaryotes are divided into kingdom Bacteria and Archaea. And we can see some of the important characteristics here, which determines which kingdom the organisms are actually placed into. So bacteria, they're unicellular. You know that their cells lack nuclei, and they lack all other membrane-bound organelles. They mostly have distinctive cell walls surrounding the plasma membrane. Some are autotrophs and some are heterotrophs. So let's just take a look at these 2 terms here. Can anyone tell me what we mean by autotrophs? So maybe students that have studied biology before may be familiar with these terms. Autotrophs, what are autotrophs? Can create their own fuel molecules, correct. Yes, so autotrophs can create their own energy molecules, their own fuel molecules. And of course, when we talk about fuel molecules, we're primarily referring to carbohydrates. All right, so they're the autotrophs. Heterotrophs, of course, cannot synthesize their own fuel molecules, so they have to ingest other animals or parts of other animals or the products of other animals in order to gain those carbohydrates. Okay, moving on to kingdom Archaea. So once again, unicellular cells lack nuclei and membrane-bound organelles, distinctive cell walls. Some are autotrophs and some are heterotrophs. All right, moving on to the eukaryotes, kingdom Protista. So these are mostly unicellular, but there are some very large multicellular organisms in this group. So think about the very large kelps, very large algae. The difference between those two. Okay, so you're asking what the difference is between these two groups here, these two kingdoms? Yes, all right, we'll come to that in the next lecture. We'll actually revisit these in more detail. Okay, so Protista, some of them have cell walls, but not all. Some are autotrophs and some are heterotrophs. Kingdom Plantae, multicellular land plants. The cells do have nuclei. They do have membrane-bound organelles. Cell walls contain cellulose. They are all autotrophs, and of course, you know, they can synthesize their own fuel molecules because of that process of photosynthesis. And the same thing applies to these autotrophs in kingdom Protista. So think about algae and also in the prokaryotes. Some of those prokaryotes also photosynthesize. And of course, Plantae, plants, of course, have very complex organ systems, tissue systems and organ systems. And this is actually one of the primary reasons that plants are quite different to algae. So this is one of the differentiating characters, because algae lack these complex organ systems. All right, fungi. Fungi are mostly multicellular, but of course, winemaking students will say, hang on, what about the yeasts? They're all single-celled, but most fungi are multicellular, and those cells actually elongate to produce these thread-like, very long, very fine thread-like cells called hyphae. And of course, think of the molds, and you'll be familiar with what this is referring to. Cells have nuclei, they have membrane enclosed organelles, cell walls contain the substance chitin, and fungi, unlike Plantae, are all heterotrophs. Heterotrophs by absorption. All right, and finally, kingdom Animalia. So they're all multicellular. Cells have nuclei, they have membrane-bound organelles. Their cells lack cell walls, so this is an important distinguishing character. They're also all heterotrophs, just like the fungi, but they're heterotrophs by ingestion, and they have complex tissue and organ systems. And we'll revisit each of these groups in turn in our very last lecture, classification of living organisms. So we're going to start with the prokaryotes, and of course, you know, bacteria are prokaryotes. They are single-celled, or mostly single-celled. Some bacteria, when they divide, the new cells actually all stay joined together. So they'll form like chains or groups of cells, you know, representing a you know, bunch of grapes, for example. So quite different in the shapes and clusters that they form. All right, bacteria mostly have semi-rigid cell walls, which is around their plasma membrane. They lack membrane-bound organelles, and their genetic material, that is their DNA or their chromosome or chromosomes, sits free in the cytoplasm. All right, bacteria are really, really diverse in terms of how they function, in terms of their size, in terms of their shape, really diverse. But it's often difficult to see this because you need at least a compound light microscope, but sometimes you even need something more powerful, like an electron microscope. Okay, so let's have a look at the relative sizes of microbes. And firstly, we'll start with a single eukaryotic cell in blue. So the size of this eukaryotic cell, this is Saccharomyces, a yeast, and we can see the dimensions there, 8.6 microns in diameter. Now let's compare that to a typical bacterium. So that's one bacterial cell there. This is E. coli, Escherichia coli, and you can see it's much smaller at 2 by 0.5 microns. Then we have Staphylococcus aureus. Everyone's probably heard of golden staph. And this one has the dimensions 0.8 microns. And then we get down to one of our very small bacteria. This is Rickettsia rickettsii, and it has these dimensions 0.4 microns. All right, so most of these can actually be resolved using a normal light microscope, which we do often in the lab when we study microbiology. You can't see them very well, but you can definitely resolve individual cells well enough to be able to pick the shape, the basic shape of the bacterium. Probably not this one, not Rickettsia rickettsii. And now let's compare those sizes to the viruses. So here we have the smallpox virus. This is considered to be a large virus, 0.2 by 0.3 microns. Influenza virus, smaller again, at 0.1 micron. Tobacco mosaic virus, this is a common virus of plants. Smaller again, and then a small virus, the polio virus, at 0.03 micron. And of course, to actually view these microbes, you would need an electron microscope. All right, the prokaryotes are divided into those two groups, bacteria and archaea, on the basis of biochemical characteristics. If we look at the level of superkingdoms, then the superkingdom bacteria was formerly known as the eubacteria, which means true bacteria. The superkingdom archaea was formerly called the archaeobacteria because they were originally thought to be the first bacteria, the first microorganisms to evolve. So they were referred to as the ancient bacteria. But it's now known that both the bacteria and the archaea seem to have evolved at around the same time. All right, so let's start with bacteria. We'll look a little bit more closely. Bacteria are really incredible in terms of the diversity of their metabolic pathways. They've been shown to basically utilise just about every energy extraction method that's thought to be theoretically possible. It's quite incredible what they can actually degrade. So it's really only, so far it's only been discovered that there's some plastics and some organochlorine compounds, so some herbicides and some pesticides that are actually resistant to bacterial degradation. So quite incredible. Characteristics of bacteria. So one of the most important characteristics of bacteria that differentiate them from the archaea is the presence of this complex compound called peptidoglycan. So this is a major component of the cell wall of bacteria, but it's not found in the archaea, in cell walls of archaea. All right, within the group known as the bacteria, there are some nitrifying species, I'll come back to that word, and some photosynthetic species. All right, let's look at one group of bacteria, one important group, the cyanobacteria. Unfortunately, cyanobacteria have been referred to for many years now as blue-green algae, which is very, very confusing for lots of people that don't realise that when you're talking about or looking at blue-green algae, you're actually looking at bacteria, not really algae. They produce a particular pigment called a phycobilin, and this is a pigment that actually gives that characteristic blue-green colour, which is obvious when they reproduce very quickly in shallow waters, often as a result of increasing temperatures, increasing nutrient concentration, which could be, you know, runoff from fertilised, over-fertilised pastures or crops, or even from sewage, as an example. So that particular pigment that they produce is a pigment that enables them to capture wavelengths of light that they wouldn't normally be able to access using their normal photosynthetic pigments. So that's the cyanobacteria. Now there's other groups of bacteria that can form what are called endospores. So these are very highly resistant dormant structures, and these spores can remain dormant in the soil, in the water for decades, for decades. They are so resistant. And these spores actually have comprised of several different layers. Each layer has high resistance to a particular type of external stress, if you like. And the main aim of those endospores is to protect the DNA, protects the DNA of the bacterium. So here are two groups of bacteria, two genera, I should say, Clostridium and Bacillus, that can produce endospores. And the reason that these are important is because many species of these endospore forming bacteria are very important pathogenic agents. So if we think about the species Clostridium tetani, that bacterium that produces endospores causes tetanus. So you can imagine how important it is to be able to destroy endospores. And here's another good example, Bacillus anthracis. That's the bacterium that causes anthrax. And that's why you can suddenly have an outbreak of anthrax when those spores that have been lying dormant in the soil for such a long time are activated to actually germinate and resume normal growth. All right, so let's revisit the archaea very briefly, and the archaea are even more specialised than the bacteria. And for this reason, they tend to occur in very specialised habitats, very extreme environments. So in the past, a few decades ago, they were actually referred to as the extreme bacteria. All right, so these extreme environments can include deep sea volcanic events that are really superheated, thermal pools, really hypersaline environments, very low pH environments, and so on. So because they can inhabit these extreme environments, they're actually grouped according to the particular environment that they're able to inhabit. So those that can inhabit hypersaline environments are called halophiles. Those that inhabit very low pH, even down to nearly zero, pH 0 environments are called acidophiles, and those that can inhabit high temperature environments are called thermophiles. So as I said earlier, that complex compound called peptidoglycan is not found in cell walls of archaea. So biochemically, they're quite different. There are actually quite a few other differences. Their ribosomes are very different to the bacteria. Their ribosomes are actually more similar to those found in animals than they are to the ribosomes found in bacteria. And there's quite a few other differences. All right, so there's no nitrifying or photosynthetic species in this kingdom. All right, so let's consider bacterial populations on the skin and inside the animals of not just humans, but other animals as well. And you can imagine that our bacterial populations are very, very large and very dense. If we look at the number of cells on the human skin, and that's nothing compared to what you would find in the gut. I mean, look at this. Look at this number per gram of human fecal material. It's incredible. And in fact, they've estimated that if you were to consider the number of bacterial cells in and on our bodies and compare that to the number of human cells, we're actually more bacterial than we are humans. All right, so bacteria, of course, can cause a lot of problems. So this particular rash that you can see here is caused by Staphylococcus aureus, so golden staph, also referred to as a superbug. And it basically, that bacteria actually produces these toxins that kill cells. Okay, and here we have one mycobacterium that causes the disease called leprosy. So it actually attacks the peripheral nerves and causes these very disfiguring lesions here. All right, we can't really talk about bacteria without at least referring to one of those very, very important biogeochemical cycles. So the way that certain nutrients move through ecosystems, whether we're talking about large ecosystems or a local ecosystem on the farm, we need to consider those important nutrients. And we'll just concentrate here a little bit on the nitrogen cycle and how the bacteria are critical within the nitrogen cycle. So let's consider on this particular slide, we could refer to this as phase one, if you like, phase one or stage one of the nitrogen cycle. And it involves these incredibly important bacteria called nitrogen fixing bacteria. So this includes the bacteria known as the cyanobacteria. It includes other bacteria that are plant symbiotes. So these are bacteria like rhizobia that are very, very closely associated with the roots of certain plant species. Then we have free-living soil bacteria belonging to the genus Clostridium and others. So these free-living, they're not associated closely with plants. So these are the nitrogen-fixing bacteria. They are the only organisms on Earth capable of taking atmospheric nitrogen and converting it into a form that can then be used by plants and by other organisms. So they're the only organisms capable of fixing molecular nitrogen. That is a huge statement. Here is the chemical reaction. Here we have our molecular nitrogen. Do legumes contain, sorry, I've got a question. Yes, absolutely. Legumes contain plant symbiotic bacteria. Absolutely. And that's why they're very commonly used in crop rotation for that important nitrogen fixing process to occur. All right, so here we have our atmospheric nitrogen, N2. What these nitrogen fixing bacteria do, they couple that elemental nitrogen, that molecular nitrogen to protons and electrons to form this polyatomic ion. And of course, you know that is the ammonium ion. All right, this reaction is sensitive to molecular oxygen, so it actually occurs in a highly reducing or anaerobic environment. All right, so what happens to the ammonium ion? So once the bacteria actually convert that atmospheric nitrogen into ammonium, they then immediately use that ammonium ion to form these two amino acids, glutamine and glutamate. And then the nitrogen is then stable and can be utilised for other applications. So really, really important process in this particular stage. All right, let's look at another stage of the nitrogen cycle. Let's just call this stage 2, and it involves other species of bacteria called nitrifying bacteria. Nitrifying bacteria will take that fixed nitrogen in the form of ammonium, and they will convert the ammonium ion either to nitrite or nitrate. So in other words, they transform that fixed nitrogen into different nitrogen forms, and different species of bacteria will do this. So from ammonium to nitrite, species belonging to this genus, Nitrosomonas, and then to nitrate, we're looking at different bacteria. So for example, those belonging to the genus Nitrobacter. They also transform ammonium that's generated from the decomposition of organisms. All right, so, and of course, you know that's important because nitrate is after ammonium, or I should say, along with the ammonium ion, nitrate is also an incredibly important form of nitrogen available for uptake by plants. All right, stage 3 of the nitrogen cycle, and this involves the denitrifying bacteria, for example, such as Pseudomonas. So denitrifying bacteria will take that nitrite and nitrate. They use it as terminal electron acceptors. Don't worry too much about that because we haven't learned about those processes yet. But what they basically do, as a result, they produce gaseous nitrous oxide and molecular nitrogen. In other words, that important fixed nitrogen is returned to the gaseous stage and released back into the atmosphere. So you can imagine that this is not something that you're after. And this is a problem with anaerobic soils, highly compacted soils, waterlogged soils, and so on. Because of course that important nitrogen is no longer available for plants and other organisms. All right, moving on now, kingdom fungi. So kingdom fungi also includes a diverse range of organisms. It includes yeasts, truffles, mushrooms, rusts, and molds. Some of the characteristics, they are all eukaryotic. They are all heterotrophs. They are, many of them are filamentous, but of course the yeasts are single-celled organisms, and they mostly have chitin in their cell walls. So here's a couple of examples. So here we can see a rust, a coffee rust, I believe this is. And here we have, I can't remember the name of this particular mushroom, but I know it is a hallucinogenic mushroom. So, and in large doses, it can be fatal. Oh, thank you. Don't ask. All right, someone has just answered that question. Amanita muscari, thank you. I won't name you, and I won't ask how you know that. All right, classification. All right, very complex, the classification, it changes, or it is changing all the time. But basically we have 6 phyla. These 4 phyla are those that can use both sexual and asexual reproduction. You don't have to remember these names. It basically includes the water molds, sac fungi, and club fungi. And here we have an example pictured here. This is Armillaria growing out of the base of a rotting tree trunk. And of course, what we're seeing here, these are just the fruiting bodies, the reproductive bodies. Most of the fungus is actually found within the actual tree itself and consists of those filamentous hyphae that we mentioned earlier. All right, a phylum here with, that have only been observed to use asexual reproduction, Deuteromycota, and then of course, phylum Mycophycota. These are the lichens. And you might know that the lichens are actually a symbiotic relationship between a fungus and a photosynthetic organism such as an algae or bacteria. Okay, so two organisms actually working together to form a lichen. Okay, I still won't ask about the Amanita muscari. All right, so fungi are the closest relatives of kingdom Animalia, but fungi, of course, have cell walls and they have chitin in their cell walls. But they, and of course, that is very different to animals because animal cells don't have cell walls. But this is another important difference too, that fungi, most of the fungal cells are actually haploid. That is, the nucleus in those cells only has one copy of each chromosome. You probably imagine that that's very different to animals, where most animal cells, that is apart from the reproductive cells, have diploid nuclei in their cells. That is 2 copies of each chromosome in each nucleus. And of course, animals lack chitin, or they lack cell walls, and of course they lack that compound called chitin. All right, so we've mentioned that fungal cells, well, that is the filamentous fungi, their cells elongate, and these are referred to as hyphae, singular hypha. Hyphae are fine tubes. The cell wall encloses, of course, the plasma membrane, which in turn encloses the cytoplasm and all the organelles. These filamentous hyphae, they grow, they elongate, and they branch at the tips, at the actual tips of the hyphae, where the wall, the cell wall, is still developing and it is still quite elastic. The main body of a fungus is called a mycelium, and the mycelium is often just a mass of hyphae. We've got a great image here, an amazing image. This would have been taken with a scanning electron microscope, and we can see here a spore, a fungal spore that has landed on the surface of a plant. The spore has germinated, and we can see these elongating cells here, that is the hyphae. And this particular hypha here has found an entry point into the plant. And this is a stomata, or rather I should say a stoma. And when the hyphae, the fungal hyphae, finds this entry point into the plant, it produces this infection structure called an aprosorium. So fantastic image there, not easy to get something like that. All right, and here we have the tip of a fungal hyphae. So let's have a look firstly at what we have inside here. So we've got, it's only showing the Golgi apparatus, not showing the other organelles. We should start with the outside. So we've got the outer cell wall, which is made of chitin. Then to the inside of that, we have the plasma membrane. Here's the growing tip. This is where the hypha is elongating. It's growing here. From the Golgi apparatus, we can see all these vesicles that are budding off. And there are 2 types of vesicles that are shown here. So in purple, we have the vesicles that are carrying digestive enzymes. These enzymes are actually secreted from the hypha, and the enzymes will actually break down any substances that are found to the outside, and then those nutrients can be taken up by the fungi. So of course they're released by that process called exocytosis, and then nutrients are taken in to the cell using the process called endocytosis. All right, the other type of vesicle that has budded off from the Golgi apparatus in green, these are vesicles that are carrying cell wall components. So they're being targeted here to this part of the plasma membrane so that the new cell wall here can be formed. Fungal nutrition. Fungi can digest nearly any form of organic carbon. They are our primary decomposers in any ecosystem. And in fact, the fungi are the only organisms that are known to be able to decompose lignin. So remember lignin, that very tough polyphenolic compound that goes into secondary cell walls. So when you're talking about decomposition of any woody tissue, you need to have fungi in that environment. All right, fungi are all heterotrophs, as you know, and most of them prefer glucose as their source of carbon. Fungi are all saprophytes, and that is, they basically release those digestive enzymes into the environment, decompose the compound, and then take up the nutrients. So that's that. And of course, there are fungi that are parasites, highly specialised fungi. All right, fungal reproduction. Fungi reproduce by spores. They form spores. And of course, a spore is just a little protective, very protective, highly protective coating, which surrounds one or several cells. And those spores are, of course, the primary means by which fungi disperse and survive. And certain changes in the environment trigger fungi to start producing those spores, that is, trigger them to start sporulation. All right, sexual spores. There's only one type of sexual spore that's produced in each species. But asexual spores, that is for those fungi that produce both sexually and asexually, they can be up to 4 types of asexual spores produced in each species. And here's another amazing image that you can see here. So we have these standard hyphae here growing on the surface of this organism, and then something has triggered this fungus to start reproducing. So it starts actually forming these upright hyphae, and these spores are actually developing, so ready to be released. Okay, now I'm afraid to ask about the name of these hallucinogenic mushrooms shown here. So some lovely images, and then on the right we have Trichophyton rubrum fungus that causes the dreaded ringworm. Okay, all silent in the chat. Can anyone tell me what these mushrooms are called? No, silence. So I have an answer, but it's a silent answer. Yes, they are commonly referred to as magic mushrooms. That's right, magic mushrooms or just shrooms. Absolutely right. All right, so we'll leave that very interesting kingdom fungi and move on to kingdom protista. The protists are all eukaryotes. Most of them are single-celled and aquatic. Most of them have little structures called flagella, well, one or more, flagella or cilia. They look like little hairs. They're not hairs. They're actually projections of the cell which enable the organisms to move around. They are very diverse. Some of the protists can photosynthesize, others are parasites, others are predators, and some gain their nutrition by absorption. Okay, here are just some groups of protists. So the choanoflagellates, marine protists, we have slime molds. The photosynthetic protists includes the red algae and green algae. We have the chromists, alveolates, and so on. You don't need to remember all these. But this is a very interesting life cycle, if you like, of a slime mold. This particular slime mold is called Dictyostelium discoidum, and it actually starts off as these single-celled organisms. They're found in moist soil, moist leaf litter and so on, and they happily go around and feed on bacteria. But then something quite amazing happens. A signal takes place, is given out. They're still not sure what that signal is, and it basically triggers all these single-celled amoebae to come together, to cluster together, to form this little slug. It's about, you know, 1 to or 2 to 4 millimeters in length. So this slug consists of thousands of those single cells all joined together. The little slugs then move off, and when they find the right place, they actually prop and they start to reassemble themselves to form these upright fruiting bodies, these upright reproductive structures. And you can see an actual image there. So these the cells that form the base and the stalk, they basically they actually die at the end of this process. The cells here, they actually form spores, and those spores are released and the life cycle starts once again. So really fascinating. And here is an actual image of one of those little slugs as it's moving along. And you can see the scale bar there, 0.5 millimeters. So quite fascinating, the slime molds. And here are some nice images from your textbook of the algae. So we've got some red algae here, green algae, here multicellular green algae, single-celled green algae down here, and the enormous brown algae that you can see here. All right, green algae. So you know, or you would know that algae have been the subject of intense research for many years now, because they're basically the scope for the use of algae in biotechnology is just enormous. And here's just a couple of examples for you. So this particular algae called Dunaliella solida is intensively farmed for this substance beta-carotene. Really important because beta-carotene is very easily converted into vitamin A. And here's one of these large ponds. This is in Western Australia, I believe. And this is where they farm Dunaliella. So if you expose Dunaliella salina to high salinity, very high salinity and very intense sunlight, then it produces lots and lots of beta-carotene. So here's another example, this Haematococcus pluvialis, which is farmed for this compound here, used as a fish food additive. And this one is fascinating. Batriococcus braunii actually produces these long chain hydrocarbons that can actually be processed to produce petrol. So really exciting possible future source of alternative fuels. All right, moving on to this group called the oomycetes, which includes water molds and downy mildews. And they're actually very similar to fungi, these organisms, but their cell walls contain cellulose rather than chitin. So you can see why they're thrown into this particular kingdom. All right, just a couple of examples here. Phytophthora cinnamoni causes this extensive dieback of trees that you can see here. Here's another famous example, Phytophthora infestans, which caused the famous Great Potato Famine in Ireland in the 1840s. And it's estimated that anywhere from 1 million to 2 million people died as a result, given that potatoes were the staple diet. Eutypa, is that like Eutypa dieback in vines? Probably not, because Eutypa, I don't know if I'm pronouncing that correctly, Eutypa doesn't normally cause, well, from what I know, doesn't cause the vines to actually die. It does cause them to be not as vigorous, and it certainly doesn't look very appealing. But my understanding is that doesn't actually kill the vines. But I could be wrong. Best that you talk to someone like Andrew Davey about that. All right, alveolates, another interesting group. Often they're referred to as plankton. Many of them are photosynthetic. Some of them cause these expansive red tides when growth is really explosive due to an influx of nutrients and temperature and so on. And some of these red tides produce some really potent toxins, which everyone's probably aware of, and those toxins can accumulate in fish, in shellfish, and so on, and they can be absolutely lethal to humans. All right, another interesting group, the apicomplexans, and the most famous member of this group is the protist called Plasmodium falciparum, because it causes that disease that still kills thousands and thousands of people every year, malaria, the disease malaria. So this protist is carried by a particular type of mosquito called the Anopheles mosquito, and it's actually transmitted to humans when the mosquito takes a blood meal from a human. The Plasmodium protist then completes part of its life cycle, actually the asexual part of its life cycle, in blood cells, red blood cells, and the liver in humans. Then it is picked up again by another mosquito that bites that human, and the Plasmodium then completes its life cycle, but it actually now reproduces sexually within the mosquito, and then it accumulates within the salivary glands, ready to be transmitted to another human. So very, very complex. And in fact, it just reminds me that many, many years ago, I worked in a lab where I actually looked after blood cells that were infected with Plasmodium. So my job was pretty simple. I had to basically check the cells, and when most of the cells were actually infected, then I had to take some of those cells and add those cells to new media to keep the Plasmodium going, because otherwise, once all the cells die in that particular sample, then the Plasmodium will also die. All right, moving on to the viruses. Seems very appropriate, doesn't it? Now, viruses are not considered to be living organisms. So we refer to them as small infectious particles, if you like, small infectious particles or small infectious entities. Why aren't they considered to be living? They're not composed of cells. They have an acellular structure. And you remember when we talk about cell theory states that all living organisms are made up of cells and the products of those cells. Well, viruses are not made of cells. Another reason why they're not considered to be living is that they cannot reproduce independently. They can only reproduce by hijacking the molecular machinery within a cell that they have invaded. And number three, viruses contain either DNA or RNA as their genetic material, but they don't contain both. All living organisms contain both DNA and RNA, but not viruses. All right, so just very quickly, viruses have quite a range of different structures. The three common ones are shown here. So we've got here a rod-shaped virus. It's actually called when we're looking at the complete virus in this way, it's actually referred to as a virion. So this would be the extracellular form of the virus. That is the way that you would find it in the environment. But the way that you would find it inside an actual infected cell, you wouldn't find this complete virion. You would just find the genetic material, that is the DNA or the RNA, inside the infected cell. All right, so that's rod-shaped. Here we have another common one. This is referred to as a naked icosahedron virion. And of course, because the shape is like that of a icosahedron, so this coat, this virus coat that you can see here, or rather the virion coat, is made up mostly of protein molecules, and that encloses the nucleic acid of the virion. And that might be, as I said, DNA or RNA. So that's referred to as a naked virion, because on the right here, we have the same sort of thing in the middle here, but this one is enclosed by a membrane. So this is called an enveloped virion, because that membrane is referred to as an envelope. Now that sounds very much like it's a cell, doesn't it? Because it's surrounded by a membrane, a biological membrane. But you've got to keep in mind where that membrane came from. That membrane was actually hijacked from the cell that was originally infected by that particular virus. And when the virus reproduces and then exits the infected cell, it takes a bit of that cell membrane with it. All right, so as fascinating as they are, that's it on viruses. So before we go on, let's take a 5-minute break. How does that sound? Sounds good? All right, I'll see you in 5. Moving on now to Kingdom Plantae, and then we'll visit Kingdom Animalia. So Kingdom Plantae, of course, includes all those photosynthetic organisms that are adapted to living on land. They were thought to have first colonised the land about 410 million years ago, beginning with very, very small, inconspicuous plants that were confined basically to wet margins of wetlands and rivers. Well, that is until they developed the characteristics that allowed them to inhabit more inland areas. Plants were thought to have arisen from green algae, in particular a group known as the chlorophytes. And this is what they have in common. So they both have the same types of chlorophyll pigments. They have chloroplasts that are very similar in structure. They both have cellulose in their cell walls, and they both utilise starch as their storage material. But keep in mind that the algae, including the chlorophytes, don't have those complex tissue systems and those complex organs that you find in plants. All right, so that transition that they made from water to land required an increase in complexity, you can say, and certain specialisations of cells and tissues, particularly with regard to two very important aspects of plant physiology, that is water balance and gas exchange. So if you think about algae, you can imagine that they don't require any special water absorbing or water transport systems because they're surrounded by water. So desiccation is not a problem for algae. In contrast, land plants inhabit environments that are often water limited and they can't move around. So this transition from water to land required structural adaptations to maintain water balance, to be able to extract water and nutrients from the soil, to be able to transport water and nutrients throughout the body of the plant, and adaptations to be able to support the plant, enable for plants to get bigger. All right, so let's look at some of the specialisations of vegetative features that were thought to have developed basically over time in that transition. So firstly, the development of the cuticle. So of course the cuticle is deposited over the entire above ground surface of the plant. It's impermeable and acts as a barrier to water loss. It also filters out quite a substantial component of harmful UV radiation. Next, we have sporopollenin. This is a really tough substance which surrounds both spores and pollen produced by plants, and it makes them very highly resistant to degradation. Okay, next, stomata. So, of course, if we're thinking about algae, they don't have to worry about stomata or any type of pore because carbon dioxide in solution can just basically diffuse readily across the cell walls of the algae and be available within algal cells for photosynthesis. But on land, of course, carbon dioxide is gaseous and it has to enter the plant through these tiny pores called stomata. Okay, vascular supply and lignin. So land plants, of course, needed to develop some internal transport system to enable the movement of water and nutrients, that is through the xylem of the vascular system, and the movement of manufactured carbohydrates through this part of the vascular system, the phloem. And then, of course, the division of labour came about when land plants developed stems, roots and leaves. All right, so let's just look at the major groups very briefly. So this group is referred to as the non-vascular plants. They're thought to have been the first plants to evolve, the first land plants to evolve. And they lack a vascular system, so they lack a true xylem and phloem, and they also lack that substance called lignin, so they don't form secondary cell walls. And there are 3 phyla here. You don't need to remember these names, but this includes the liverworts, hornworts, and mosses. And you can see them pictured here. All right, the first group of vascular plants, so those plants that in fact developed a true vascular system consisting of the xylem and phloem, but these plants produce spores rather than seeds. And this includes the club mosses and quillworts and so on, and of course everyone's familiar with the ferns. Okay, and next we have the probably the most recent group of plants to evolve, the seed-producing plants. And the seed plants are very broadly divided into 2 groups. So we've got the gymnosperms, which are the non-flowering plants, and then the more abundant angiosperms, the flowering plants. So they all produce seeds, and of course the production of seeds, the development of seeds, was also a very important evolutionary event. All right, so the gymnosperms, non-flowering plants, so they don't produce flowers, they do bear their seeds in cones. So this includes 4 phyla, the conifers, the cycads, and we have a cycad pictured here and a cone of a cycad. Then we've got just 1 living species of ginkgo and the neophytes. Then we come to the all-important angiosperms, the flowering plants. They belong to phylum Magnoliophyta. There's over 250,000 species of flowering plants, and these are our major producers in terrestrial ecosystems, providing food, timber, fibres, drugs, and so on that sustain the human population. Okay, everyone's familiar now with the concept of secondary growth, and secondary growth allows plants to become large. So remember, we're talking about that woody growth, and that arises from the presence of the vascular cambium, which produces woody tissue, and we won't go over that again, but that's shown there for your interest. All right, so let's talk a bit more about flowering plants. The flowering plants are broadly divided into 2 groups, the monocots, which belong to class Liliopsida, and the dicots, which belong to this class here. So let's have a look at the differences between the monocots and the dicots. And we'll probably look at that, yes, better here on this slide. So let's start with the monocots, and we can see these characters here in the top panel. So the monocots have 1 cotyledon, that is, they form 1 leaf inside the seed. 1 cotyledon, 1 seed leaf, it's referred to. It's not a true leaf. It is referred to as a seed leaf. And when the seed germinates, only 1 leaf initially appears, or 1 cotyledon appears. All right, if we're looking at a cross-section of a stem of a monocot, you would see that the vascular bundles, these little patches here, they're scattered more or less randomly throughout the stem. In contrast, you've seen an image like this one, where in dicots, the vascular bundles are arranged in a circular arrangement within the actual stem. Venation, leaf venation. So the veins in leaves of monocots tend to be found as parallel veins. And if we're looking at the flowers of monocots, the flower parts, not just the flower petals, but the flower parts are found in multiples of 3. So let's contrast that to the dicots. So dicots produce 2 seed leaves within the seed, and that's how it emerges when the seed germinates, has 2 cotyledons. We've looked at that one. Venation in the leaves, so in dicots you will have branching veins, also referred to as reticulate venation. And then if we look at flower parts, in dicots, the flower parts are found in multiples of 4 or 5. So some really good pointers there to distinguish monocots from dicots. Okay, also important within flowering plants. So if we look at just a single flower here, and of course you're probably already familiar with the fact that many flowers have both male and female reproductive parts. So we've got this central stalk here. This is the carpel, that's the female reproductive part. And in this case, it's surrounded by multiple fine stalks, which are the stamens. And they are the male reproductive parts of the flower. So if we take a closer look, so this is part of the male reproductive part. It's called the anther, and this is where pollen is produced. Here we have the female reproductive part here enlarged, and within this carpel, we have this ovary, which contains an ovule, and within the ovule, we have the egg cell. Now, if that's fertilised by the sperm cells found within the actual pollen, then an embryo starts to form. That embryo, if we look over here, so in green, we've got the embryo that's forming, and that is that's surrounded by a seed coat, which starts to develop around that embryo. And what was originally the ovary, here's the seed coat here. This whitish part here is the seed coat. And what was originally the ovary starts to form a fruit on the outside. So when we thinking about the fruit, think about the fleshy part of a grape or a peach, for example. Okay, and of course the fruit is designed to protect the seed and to aid in its dispersal by fruit-eating animals. We've got different types of fruits, simple fruits which are derived from the ovary of a single flower, such as in grape, apple and peas. We have aggregate fruits derived from clusters of those female carpels that are found in a single flower, for example, raspberries and blackberries, and then multiple fruits which are derived from clusters of carpels from several flowers, for example, pineapples. And our final kingdom, Kingdom Animalia. So what is an animal? There are actually several different characters associated with animals, but very important, they are all multicellular. There is always a division of labour with specialised cells, and in all animals, apart from the sponges, cells will form tissues and organs. They all undergo embryonic development from a single cell, which is called a zygote. So this is a single fertilised cell, and they are all heterotrophs. So these are just some of the characters that define animals. All right, there are 2 subkingdoms under Kingdom Animalia. One is subkingdom Parazoa, which just includes the sponges. All other animals are placed into subkingdom Eumetazoa. Eumetazoa is further divided here into 2 groups: the animals that exhibit radial symmetry and animals that exhibit bilateral symmetry. So let's just take a look at that. Where's my pen? So all that means when we're talking about radial symmetry, so if we think about, if we're looking at the top of a jellyfish, for example, we can actually divide that into any plane. So we can take this plane and produce two similar halves, or we can divide it in this plane. So any plane will produce two similar halves that are mirror images of each other. But if we consider bilateral symmetry, and this is where the drawing starts to really lack. All right, don't ask me what sort of animal I'm drawing here, but there you go. That's supposed to be an animal with bilateral symmetry, and you can only divide this animal in one plane to produce two similar halves that are more or less mirror images of each other. And that's what we mean by bilateral symmetry. So I'll just clear that awful drawing. All right, so the bilaterally symmetrical animals are further divided into two groups. Here we have the protostomes and the deuterostomes. And let's look at what they are, what that means. I'll just get the pen once again. So very early on in the development of the embryo, of the animal embryo, a little pocket develops like this. Okay, so this is a mass of cells. Little pocket, this little opening has developed here. And if that little opening eventually develops into the mouth of the animal, okay, then it is classified as a protostome. If it develops eventually into the anus of the animal, then those animals are classified as deuterostomes. So it's as simple as that, but that is used for classification purposes. All right, so here we have one of those phylogenetic trees of the animal kingdom. And you can see firstly radial symmetry. Okay, these animals here display radial symmetry. All the others display bilateral symmetry. And within this group here, they're further divided into the protostomes. And then over here, we have the deuterostomes. All right, so let's run through just some of the different groups of animals, starting with the sponges. Probably the simplest of the animals. They belong to phylum Porifera. These are aquatic animals with the different cell layers that all animals display early in embryonic development, but those cell layers are never organised into tissues or organs. Then we have the phylum Cnidaria, which includes the polyps and medusae. These are radially symmetrical animals with only two tissue layers in the body wall. This includes jellyfish, corals, and sea anemones. Okay, our next group are the flatworms, phylum Platyhelminths, and this includes both free-living and parasitic flatworms, but also flukes and tapeworms. And we can see the life cycle here of the liver fluke, which spends part of its time in this case, in a sheep, and then part of its life cycle in a particular species of snail. Next, we have the segmented worms, which belong to phylum Annelida. Segmented worms include marine worms, earthworms, and leeches, and they're categorised by the presence of what's called metameric segmentation. And you can probably see in this earthworm all those multiple segments in its body, and those multiple segments are referred to as metameric segmentation. Within each segment, you will see a repetition of functional units, that is repetition of clusters of nerves, repetition of reproductive structures, and so on. Next, we have phylum Mollusca. This includes clams, mussels, snails, slugs, octopus, and squid. And here are a few different characteristics. Most of these organisms don't have all of these characteristics, but they certainly are quite common. And we can see those characters here in this generalised image of a mollusk. Next, phylum Nematoda. These are the roundworms, and they can be free-living or endoparasitic. They are very, very successful with over 25,000 species. And just about every species of plant or animal that's been studied has found to be associated with a nematode, at least one species of nematode. Next, we have phylum Arthropoda, which includes spiders, crabs, millipedes, crabs again, yabbies, and insects. And here are the main characteristics of the arthropods. They have jointed limbs. They have a chitin containing exoskeleton. They also exhibit metameric segmentation, but with a difference, because that's metameric segmentation comes with tagmatization, which is when some of those segments are actually clustered into functional regions called tagmata, and those functional regions, for example, could be the thorax or the abdomen. Okay, next we have phylum Echinodermata, which includes the sea stars, sea cucumbers, and so on. Then we move on to phylum Chordata, acorn worms, sea squirts, lancelets, and vertebrates. They all exhibit bilateral symmetry, and here are the three important characteristics of the chordates. The presence of a notochord, the presence of pharyngeal slits, and the presence of a dorsal hollow nerve cord. And we'll look at those characters again here. All right, so what is the notochord? It's a dorsal rod which is situated between the nerve cord, so think of the spinal nerve cord and the gut. So that's the notochord. Then we have the pharyngeal slits, which are simply paired openings in the pharynx. By some chordates, they're used for filter feeding, so think of fish. And of course, in humans, pharyngeal slits eventually form into other parts of the body, such as parts of the jaw and parts of the thymus gland, and so on. All right, the third important characteristic is the presence of a dorsal nerve cord. So this is a hollow nerve cord that's situated above that notochord, and it expands at one end to form a brain in some chordates. All right, and those three features are pictured here. So we're looking at a human embryo. In green, we can see the pharyngeal slits. In brown, we can see the notochord. So in humans, that is replaced. That doesn't persist, but in some of the chordates, it persists. And then we have the dorsal nerve cord in blue, and it's expanding here to form a brain. Okay, let's look at subphylum Craniata. This includes the jawless fish and the vertebrates. Characteristics within this subphylum, the presence of a head with a cranium or skull made of cartilage or bone, and a brain with cranial nerves. All right, so here we have the jawless fish, which includes the lampreys and hagfish. Characteristics, they have a cartilaginous skeleton. The notochord in this group actually persists in the adults, and they lack jaws. Okay, now let's move on to the vertebrates, which includes fish, amphibians, birds, reptiles, and mammals. So characteristics of the vertebrates, So in the adult, the notochord, as we said earlier, is replaced, and it's replaced by vertebrae. Projections from the vertebrae actually protect the nerve cord. The presence, another characteristic is the presence of these very unique cells, which basically detach. This is within the embryo, within the developing embryo. These cells, called neural crest cells, will actually detach and move throughout the embryo to their final resting place, and they give rise to various structures within the organism. Another characteristic, dentine and enamel, which often form teeth or denticles. All right, we're going to look just at the mammals a little bit more closely to finish off. So class Mammalia. Just two of the characteristics are shown here. There are a few different characteristics. The presence of epidermal hair and milk production from mammary glands. There are two subclasses as shown here. Subclass Prototheria, which includes one order, the monotremes, and subclass Theria, which includes the marsupials and the true placental mammals. So let's look very briefly at the monotremes, which are in Prototheria. These are the only egg-laying mammals, of course, and there are only 3 species, the platypus and 2 species of echidna. And these represent the most ancient lineage of mammals. Okay, and then we move on to Metatheria, the marsupials. 272 species, mostly in Australia. So this is the dominant group in Australia. And this includes the quolls, bilbies, opossums, koalas, kangaroos, and bandicoots. And the marsupials are characterised by birth of their young at a stage which is equivalent to the fetal stage in the eutherian mammals. So they're born at a very premature stage, and you can actually see that here. So this is the young of a kangaroo that's already been born, and it's actually finding its way to the pouch, which is called a marsupium. All right, the marsupials, very oddly, the females have a left and right uterus and 2 separate vaginal canals. The male of the species has a penis that is bifurcate, that is, it's forked. All right, finally, the Eutheria. So these are our true placental mammals. And there are over 4,000 species, and they are thought to have evolved 75 to 45 million years ago. So characteristics, obviously they don't lay eggs. They have a very specific type of placenta called a chorioallantoid placenta. The females only have a single uterus and single vaginal canal, and the male has a penis that is not bifurcated. And finally, our very last slide for living systems, and I'll leave you with this phylogenetic tree of the eutherian mammals. So that is it. Well done. You have completed all the lectures for Living Systems 1. Well done. And of course, it wouldn't be the same. Hooray. Yes, thank you, everyone. It's been terrific, and you guys have been really, really awesome. Your attention, your questions, it's been really fantastic. And of course, we can't finish without another set of tutorial questions, but these are not assessed, well, not for the purpose of, you know, online quizzes. So don't forget to have a go at them after you've finished all your assessments this week. Please don't forget, if you could spend a few minutes completing the surveys for each of your subjects. we would really, really appreciate it. Like I said, will only take you a few minutes. Check your La Trobe emails. That's how you get the link to the survey. There's only a few questions, and all you have to click on is basically a rating of 1 to 5 to answer each of those questions. So really, really quick. That would be great. And I think they close at the end of this week. All right, don't forget the exam timetable has been released. So have a look at your Bachelor of Ag and Tech Moodle site for this, for the exam schedule. Okay, so you all know by now that Living Systems is first one off the rank on Monday, June the 15th. Okay, so there they are. I'll put this back up if you like when I stop the recording in case you have any questions. And subject learning guide. So your last assess tutorial was due tonight. So hopefully you all got that in on time. And your last prac report, your second prac report is due this Friday by 5 p.m. So of course that's on the acid base titration. All right, fantastic everyone. I'm going to stop the recording now and I'll hang around for a while. So I'll just stop the recording. Thank you everyone. Okay, recording, stop, yes.